Yeah, this receptor has its own function. Usually what happens is the receptor is a key molecule that receives signals from the environment and then translates those signals into some type of phenotype in the cell. Receptors are also important for helping cells move. They will recognize there are actually other proteins which bind to those receptors, and the cell recognizes these other proteins and then moves towards them, so they're important for that as well. So the CCR5 receptor. So HIV uses 3 receptors. It uses the primary receptor is the CD4 receptor. That's an absolutely essential receptor. CCR5 is a chemokine receptor. It's not essential. There about 1% of human beings, 1% of Caucasians have a deletion in the CCR5 gene, which makes it so they don't produce functional CCR5, and they are resistant to HIV. I haven't found that mutation in Africans. I don't know about Asians. So you could imagine a situation where if we could figure out a way to delete CCR5 from the human genome, again, this is another strategy that could make people resistant. But HIV doesn't just use CCR5, it also uses CXCR4. And so CXCR4, as far as I know, there are no deletions or polymorphisms in the human population. CXCR4 really is an essential gene. But yeah, these receptors have their own function. HIV really evolved to be able to use them.
So your TRIM5 alpha data is really money, and like it's completely ridiculously good. And so I'm wondering, when you come up with, I mean, that's cell culture data. So what, I assume you published it, but what comes of that in terms of moving on it in some kind of useful, good, healthy, what can we do with this to?
Yeah. Now this is a really frustrating thing. This is what I'm really think about, at night and try to, I mean, I've thought about this for many years, and it's the frustrating thing is that really without gene therapy, it's difficult to use TRIM5. Now, I think that TRIM5 is involved in other aspects of the immune system, and I believe that TRIM5 may be important for an antibody-mediated response to HIV, something that could be important in developing a vaccine. But yes, you're right. The way in which you use TRIM5 is I told you that humans have this one residue on TRIM5, which really decreases its antiviral activity. You could imagine developing a drug that it's a positively charged amino acid, developing a drug that neutralizes that amino acid, removes its charge, something like that, and that would then activate TRIM5. I also think there are ways. I was just talking to somebody just a couple of days ago. We're developing technologies that are enabling us to go in and just delete small parts of the genome. So I think that we could have a technology that allows us just to delete that amino acid from TRIM5. And so maybe, we will use another virus, and vaccinate. You might not need to get every cell in your body to have that deletion TRIM5. What if you could just get 50% of your T cells in your body? So we might be able just to draw blood from people, take those cells, delete that amino acid, and then put those T cells back into people. Now, if you had a pool of 50% of your T cells that were completely resistant to HIV, you might be able just to kill off the rest of your T cells, and then those resistant T cells come and they repopulate your immune system, and now you're healthy and resistant. Now, is that realistic in the next few years? No. And is it? Yeah. And should we even be thinking about those therapies, given that given that it's really Africa where that's not going to be a viable therapy in a place like Africa, because without the healthcare infrastructure, it would be too costly, just impossible. So, I don't know. I gotta tell you, these are dark days for the HIV field. I think people are really stuck thinking about, how to make a vaccine. And
Well, I think my question a little bit more basically was, you get really bomb-ass data like you have, and awesome, you publish in Science or Nature, great, you have your Science or Nature paper. Do you just plunk it in Science and Nature and then like wait for the call to come in from a pharmaceutical company saying, hey,
no. I mean,
is it up to you to then go to them and be like, we want to do something with this?
Yeah, I mean, it's the way the pharmaceutical industry works right now is that they tend to come in at a later stage. I mean, they're much less willing these days to take risks. So there's been a lot of work done on TRIM5 over the last few years by lots and lots of labs, and I think that and a lot of work to try to develop it into a therapy, something that could get pharmaceutical companies interested. I think we already have. I mean, one of the problems that you have is that you've got a really good set of HIV drugs right now. I mean, in many ways, we don't need another drug. We really need a vaccine. And so I think that there's a lot of effort to try to understand how these innate immune defenses like TRIM5 translate into bolstering the adaptive immune system. And the adaptive immune system is really critical for making a vaccine. So I look at it. I tend to think less about how to use TRIM as a drug, because I think we've got good drugs. I like it more in terms of what it might do to increase the antibody response, increase the ability of us to make a vaccine. I mean, that's the way I really think about it. Now, there's really not that many pharmaceutical companies out there working on making an HIV vaccine because it's so risky. There's been so much failure, and it's not going to make pharmaceutical companies that much money to produce an HIV vaccine. They make a lot more money from the HIV drugs than they do the vaccine. So it's not, the vaccine is going to be made probably, in a university research lab. It's not going to be made a lab. It's going to come from a huge effort from lots and lots of labs and serendipitous discovery, which helps us, move it forward. So.
I have a question about, since HIV has such a high mutation rate, does a primary strain basically block out other strains once it's mutating, or can someone be multiply infected with 2 African versions and 1 Chinese version, that sort of thing?
Yeah, no, that's a good question. I'm not 100% sure. I believe that one variant, I think you have in your body, you have a spectrum of variants that are replicated. But I think that it probably looks like a Gaussian curve. I think there's some variants which have a higher fitness level, which tend to take over. Now, you throw drugs at those, or the immune system responds to those. Maybe those decrease when a different type of variant kind of evolves. But people can be infected with 2 different subtypes of HIV, like a subtype A and a subtype B or something like that. So certainly, you can be infected with multiple subtypes. You probably, I think it varies from individual to individual depending on your own immune system, how you fight the virus. But you can definitely have multiple variants.
Are we seeing that in the real world, or does the antibody response from fighting the first variant act sort of like a pet against the second variant, or? Are we seeing this in the lab or in people?
No, I think we're seeing this a lot in people. I mean, people will, early on in infection, during the early stages when you first get infected with HIV, you will mount both an antibody response and also a cytotoxic T cell response. These are T cells that go out and they find viral infected cells and they kill them. That response is pretty good because viral load goes way down. And then you maintain low viral load for a long period of time. It sort of slowly creeps up until you get AIDS and then you die. So we're able to mount a response. And we're able to contain virus for a little while. But then the virus continues to replicate, continues to evolve, and new variants